Okay, very good morning, my dear friends. So this is Dr.PADALA Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal, convener of CPGET PG Entrance Online Free Coaching. So today's our eminent resource person in the field of plant sciences and plant biotechnology. None of the other than Dr.O.Padmaja Madam Garu, Assistant Professor, Department of Botany, Tara Government Degree and PG College Autonomous. Madam, please welcome and share your screen. Start your session, madam. Please welcome.
Okay, thank you, sir. Yeah, good afternoon to students. Yeah, good afternoon. Myself, Dr.Padmaja, Assistant Professor of Botany, Tara Degree College, Reddy. Today, we are going to start a topic related to embryology of endosperm. These are the content I am going to discuss today. Endosperm, how the endosperm is formed and types of endosperms, functions, polyembryony, types of polyembryony, and in this conclusion, how these endosperms and the polyembryony is related in the present plant hybridization. So, when we are entering into our class, a brief idea regarding the formation of endosperm, how the endosperm is formed. So, by the one of the male gamete in the pollen tube unite with the polar nuclei, two polar nuclei. As a result, endosperm is formed. This is 3N or 3X in number. That means by the triple fusion, and the end product is known as the endosperm. Now, endosperm, and what is the function of endosperm and where this endosperm is present? Endosperm is the nutritive tissue. That means the tissue is giving nourishment for the developing embryo. That means in the embryo sac, already the male gamete fuses with the egg. As a result, embryo is formed. Another male gamete fuses with the polar nucleus. As a result, a 3N structure endosperm is formed. This endosperm is provide nourishment or giving food for the developing embryo. In gymnosperms, the endosperm is haploid. That means it is formed before fertilization. In these phanerogams, the plants are divided into gymnosperms and angiosperms. The gymnosperms are primitive when compared to the angiosperms. In angiosperm, the endosperm is triploid as it is formed after fertilization by the triple fusion. And what are the types of endosperms? Mainly, they are of different types of endosperms are there. First one is nuclear endosperm, cellular endosperm, and helobial endosperm, and ruminant endosperm. That means depending upon the topic itself, nuclear means the endosperm nucleus is dividing by the mitotic division, giving rise to a structure of a nuclear structure. That means if we see the endosperm, the number of nucleus are present in the endosperm. And if you see the cellular endosperm, as the nucleus is dividing, there is also a wall formation is taking place. As a result, they are looking like a cells in the endosperm. So when we see under the microscope, it is looking like a cellular like structure. And helobial is considered as the intermediate between the nuclear endosperm and the cellular endosperm. Now, nuclear endosperm. Mainly, the most 56% of families of angiosperms are consisting of a nuclear endosperm. And the primary endosperm nucleus means pen is divides rapidly without the wall formation. Just the main difference between the nuclear endosperm and the cellular is here there won't be any wall formation. Just nucleus whatever the kendrakalu evayithe unnayo, that will be divided and giving rise to the number of nucleus without formation of the wall. So all they remains like a free nucleate. అనేకమైన న్యూక్లియస్ అన్ని కూడా మనకు అందులో కనిపించడం జరుగుతూ ఉంటది. A central vacuole appears and pushes the nucleate to the periphery. And later, wall formation starts from the periphery towards the center. Eventually, the entire endosperm become the cellular latter wards. So, for example, if you take the coccus nucifera, in that we already know that liquid endosperm, కొబ్బరిని మన liquid endosperm అంటాము, and కొబ్బరిని అన్నమాట మనము nuclear endosperm, that is come under example. So the number of nucleus formation will be takes place in this, and the example is coccus nucifera. And the cellular endosperm, in this 25% families of angiosperms, they are of cellular endosperm. And in this type, the primary endosperm nucleus divides transversely to form two unequal chambers. That means they are micropylar chamber and the chalazal chamber. And this nucleus will divide at the same time. Wall formation will also be taking place. As a result, the endosperm will looking like a cells-like manner. So the name itself is the cellular endosperm. Example: balsam, datura, and petunia. And then the third one is the helobial endosperm. That is the different. It is an intermediate. Ante it sagamemo manaki cellular chupistundi, sagamem nuclear chupistundi. So it is considered as a helobial endosperm. It is restricted largely to the monocotyledons in order helobials. That means in the order helobials present in monocots. Mostly helobial type of endosperms are seen in monocots. Endosperm is the intermediate type between the cellular and the nuclear endosperm. In this type of primary endosperm, nucleus moves towards chalazal end of the embryo sac here and divides to form a large micropylar chamber and the chalazal chamber. In the micropylar chamber, several free nuclear divisions and the wall formation will be taking place. As a result, the chalazal chambers remain coelocytic, means many nucleus are present by the formation of the wall. And this is seen in Asphodellus. And the important one is the ruminate endosperm. We may expect the bit regarding to the ruminate endosperm. These are mainly seen in the families of Anonaceae, Rutaceae, Rubiaceae, and Myristicaceae. In these families, the endosperm is known as a ruminate endosperm. That means the endosperm with uneven irregularities on the surface. So that type of endosperm, that means the endosperm is just like a eaten or unequal surfaces will be seen. And for example, if you want to clearly understand the ruminate endosperm, if we cut the seeds of the custard apple, in that seeds we can clearly see that the ruminate type of endosperms. So the seeds like Anonaceae, Rutaceae, Rubiaceae, rumination is caused by the activity of the seed coat. And these are the diagrammatic representation of the types of endosperm. We already studied that there are different types of endosperms are there, and they are, first one is the nuclear, second cellular, and helobial and ruminate endosperm. In this, if you see the first diagram, here the black color dot is showing the embryo, and the middle center one is considered as the after triple fusion, the end product is known as the primary endosperm nucleus. And this nucleus will undergo mitotic division, and it undergoing number of free nuclear divisions. As a result, it is all the nucleus are present in the embryo sac, and so it is considered as the nuclear type of endosperm. So all the nucleus are there, and later on again the wall formation will be seen. But first we are seeing the division of the nucleus. So this we come under the coccus nucifera, this type of endosperm we are seeing. And cellular type of endosperm. Here the nucleus will divide. At the same time, the wall formation is also taking place. So like this, it is resembling like a cells in the endosperm. So the name itself is the cellular endosperm. And next one is the helobial. It is mainly seen in the monocots of the monocots. In this, the nucleus will divide, and one nucleus will move to the micropylar end and up to the chalazal end. We can clearly see that the primary endosperm nucleus is dividing, and one is moving towards the micropylar chamber and another towards the chalazal chamber. And here the nucleus that is present in the chalazal chamber, there is a wall formation is taking place. And the nucleus towards the micropylar chamber is dividing to give rise to free nucleus. As a result, it is showing the some part as a nuclear and some part as a cellular. So it is considered as an intermediate type of endosperm. And the important one is the ruminant endosperm. This is mainly we see in the Anonaceae family, and we can also see the example of Erica ketachu. Wakalo kuda manam ilanti type of ruminant endosperm we are clearly seen in this. And what are the functions of this endosperm? It is mainly, it plays a critical role in supplying nutrients for the embryonic growth. Embryonic growth means the embryo is present in the embryo sac. This embryo have to divide, redifferentiated, and it have to give a plant. For this plant, the food materials is given by this endosperm. And controlling the growth rate of embryo during the germination and the seed development. And this embryo, the seedling will be taking place, and this seedling, the seed coat have to rupture, and the enzymes have to release, and the germination have to taking place, and the seed development all be taking place. For this, the food is essential. This food is given by the endosperm that is present in the embryo sac, providing some amount of protection for the embryo. It not only showing the role of nutrition, it also providing the food for the developing embryo, and it is also giving the protection. However, the most important role in plant is as the baby plant produce source. So in order to get a small plant from a seed, that is providing the food materials for developing a small plant. And these are the related to the endosperm. Now, another topic is polyembryony. We already know that embryo, the fusion of male and female gamete that is released to the fertilization as a syngamy, the embryo is formed. Poly means many. In sometimes more number of embryos are formed. That condition is known as the polyembryony. And these we are also seeing some members. So normally a single embryo is present in each seed, but sometimes more than one embryos are present in the seed. This condition is called as the polyembryony. And it was first reported by Anton von Leeuwenhoek in the seed of oranges that belongs to the citrus family. He observed that a more than one embryo is seen. That condition is known as polyembryony. He is first reported in the oranges and the citrus. So polyembryony is mainly of two types, namely false polyembryony and the true polyembryony. False polyembryony means development of embryo from more than one embryo sac in the same ovule, which is a rare feature. That means here the embryos are formed more than one embryo sac. That means other than this embryo, there are number of cells are present in the embryo sac. So depending development of this embryo from more than one embryo sac in the same ovule, it is a rare feature. It mainly seen in Casuarina. And next one is the true polyembryony. Development of more than one embryo within the same embryo sac. That is called as the true polyembryo. That means if the embryo is giving rise to the another embryo that is called true polyembryony. If the other than embryo, their number of cells are there in the embryo sac or from the outside the embryo sac, that may be considered as the false polyembryony. And it is following types. They are cleavage polyembryony. So cleavage means the embryo is dividing. That is known as cleavage polyembryony. It is more common in gymnosperms but rare in angiosperms. So this type of cleavage polyembryony is seen in gymnosperms. In this type, the zygote or proembryo switch into more units or develop into embryos. That means the zygote or the embryo may be dividing and giving rise to more than one embryo. It is come under the true polyembryony, and it is also the embryo is dividing, so it is called as the cleavage polyembryony. Embryos are formed from zygotic embryo by the budding. That means the embryo is giving rise by the division. As a result, if you see, it looks like a budding. And here we are seeing the how the polyembryo condition is seen here. And in zygotes divides irregularly to form a mass of cells. Sometimes the zygote is also dividing, and each nuclei of these embryos may be developed into the embryo. So it looking like a more number of embryos are seen here. So of which this lies toward the chelazal and grows simultaneously and give rise to many embryos. That means in the chelazal side, the embryo will be dividing and giving rise to mass of embryoids, and these embryos may be developed into the embryos. For example, in our tissue culture, we are giving the hormones in order to develop from the callus, we are getting the embryos. But in this natural condition, the zygote itself is dividing and giving rise to the embryos. So such condition is called as the cleavage polyembryony. And sometimes the embryos may be developed from the synergids, sometimes may be developed from the antipodal, sometimes they may be developed from the integuments, or they may be developed from the nucellus. So this condition, depending upon the development of embryos from the cells, they are of different types. If the zygote itself and divides to give rise to the embryos, that is called as the cleavage polyembryony. And this is the diagram showing the cleavage polyembryony. And the embryos from synergids. In this type, additional embryos from one or more synergids. In Argemone mexicanum, these are the medicinal plant, and Phaseolus vulgaris, embryos develop from unfertilized synergids since they are haploid in nature. We already know that the synergids or the haploid in condition, if they may be developed into embryos, that are considered as the haploid embryos. They are formed from the synergids. In Sagittarius and Astrolocacia, the embryos develop from the fertilized synergids and they are diploid in nature. Before they may be formed, they are haploid in nature, and after fertilization, they are formed, they may be a diploid in condition. And embryos from the antipodals. We already know that after fertilization, the antipodals may be disappear, but sometimes they may be short-lived and disintegrated before or after fertilization. But very rarely, the antipodal cells become egg-like and divide to form the pro-embryos. And these are the diagrammatic representation of the embryos forming from the zygote and the embryos forming from the antipodal embryos. Here, in the first diagram, we are showing that the zygote is dividing and the cleavage polyembryony we are clearly seeing, and that are develops into the embryos. And in the antipodals are also may be developing and giving rise to the embryos. So the formation may be the antipodal embryos. And next one is adventive polyembryony. In this, adventive embryos formed from the nucellus. So it is an important bit is from which cell of that adventive polyembryony is formed is that may be nucellus or the integuments. So if the embryos are formed from the nucellus or integuments, that type of polyembryony is called as the adventive polyembryony. It is very common in citrus and Magnolia indica. And this is the diagram we are seeing in the ovule. So these are the integuments. Inside the integuments, the tissue is known as the nucellus. It is always diploid in condition. And the nucellus is also giving rise to developing into the embryos. So this is come under the adventive polyembryony. And the embryos that are formed from the nucellus is always diploid in condition. So sometimes this type of embryos are also will be seen here. And next one, what are the causes for this polyembryony? Why this type of abnormal condition we are seeing? This is due to the some death of the cells or due to the some hormones and due to some cells will also releases some hormone-like structures. These are also may be related to this polyembryony. So this, according to some Herberlon, he said that due to necrohormone theory. This theory was proposed by Herberlon. The theory states that disintegrating nucellus cells. That means the nucellus, a mass of nucellus cells are present. Ovule. And these nucellus cells will sometime will be disintegrate. And these disintegrating nucellus cells will helpful or will stimulate the adjacent cells to releasing this some type of necrohormones. These cells stimulates and they may be helpful for the adjacent cells to divide and form the adventive embryos. So like this, they are stimulating some hormone-like structures. So it is called as a necrohormone theory. According to this theory, the sum of the disintegrating cells, they are releasing some hormones. This may be influence the some of the cells to develop into the embryos. And next one is the physiological theory. That may be due to some physiological changes. In every plant, we are seeing some physiological changes in the from the seed to the death of the plant. So this theory was proposed by Prof. Prof states that the embryo numbering in citrus seeds is influenced by the following factors. He said that age of tree. Depending upon the aging of the plant, the number of hormonal imbalance will also be taking place, and in some time in vegetative state, some type of hormones will be stimulated, and in the flowering stage, some type of hormones will be stimulating, and at the time of leaves shedding or at the time of fruit formation, this hormones maybe will be related. So depending upon the age of the tree, also this type of condition we are seeing, and a high yielding capacity and the nutritional status of the plant and the orientation of branches of the trees. These are all due to some physiological changes. We may see this type of condition of formation of more number of embryos. So this condition is called as a polyembryony. But nowadays, more in horticulture or in floriculture or whatever this floriculture research are going on, this depending upon the taking these embryos and developing into the new varieties or due to the hybridization or hybrids. And what is the significance? And it plays an important role in plant breeding and the horticulture. and the clones obtained from the repeated vegetative propagation. By this vegetative propagation, the new varieties of the plants will be getting, and the desired varieties are also will be getting, that will be having the sustainable capacity, and that will be having the resistant capacity and for the future environment. And advent to embryos are also for significance in morphogenetic studies. In order to study the different types of characters or different types of morphological characters, these advantage to embryos is very, very helpful in the research work. And the overall part, if we see the is the main concept of regarding our topic today is the endosperm and the polyembryony. The endosperm and embryo are important parts of a seed and responsible for the germination. If you think that if the seed is not having a quality embryo, and that may be not leading for the seedlings, or that may be not leading for a good crop, so there won't be any yielding will be seen here. The seeds may be of viability, seeds will not be produced. So a good quality of embryo will be there. This embryo may have to divide and give rise to the seedling. For that total journey, the endosperm is providing the food for the developing embryos. And endosperm in the seed is developed due to the triple fusion of a male gamete with the central cell of the embryo sac. So today's in our topic, we completed the endosperm polyembryony, and the endosperm main function is the nutritive function. There are different types of endosperms are there, and the polyembryony means more than embryo formation, more than one embryo. That condition is called as polyembryony. And in this false polyembryony is a true polyembryony is there. In the true polyembryony, this cleavage polyembryony and the adventive polyembryony having a more importance, significance in the hybridization and the horticulture.
Madam.
Sir.
Madam, thank you very much.
Thank you,
really, sir. Your presentation, especially.
Madam, Madam, share this class.
Some of the participants have queries. Okay, please ask your respected madam.
Santosh only. Evarana maatladedi? Prakash.
Madam, share this class in group na.
Sir will send na.
Okay, ma'am. Thank you.
Okay, definitely. Friends. Also, and also some of the PDFs also.
Sir, your voice is not audible.
1000.